Today's topic is enzymes. So let's talk about the enzymes now. Enzymes are described as biological catalyst. So what is meant by biological catalyst? Catalyst is something that increases the rate of a chemical reaction and it is not changed by the reaction. So this is the first thing that you need to know in this topic, that they are biological catalysts. Then there are certain enzymes which have some properties, like they are protein in nature, they are very specific, they can be denatured at high temperatures, they are found in the living cells or they are formed by the living cells particularly, and a small amount of enzyme can speed up a reaction to its double. So lock and key hypothesis explains the mechanism of action of enzyme. How these enzymes work basically. You can see that enzyme is just like a lock which can bind to a very specific key. As a lock can only recognize a specific key, similarly, an enzyme can recognize only a specific substrate, and that specific substrate has to be complementary to the shape of the active site of enzyme. When a substrate binds to the enzyme, then the next step is the formation of the products. The enzyme remains the same, whereas the product is formed at the end of the reaction. So, enzyme can also be used to break down or to make up the products. So, reactants can be taken in the form of these green boxes and they can be joined by another enzyme. So, we have discussed here two types of enzymes. Now, let's discuss their examples. One enzyme is particularly to break down the structure of the starch, and that is present in our mouth, and it is called as amylase. Amylase breaks down the starch molecules, the long complex starch molecule, into maltose by this amylase enzyme. Okay, now the maltose is going to be converted into glucose molecules by maltase enzyme. So this is where the enzymes are broken down. In living cells, we can find lipase, carbohydrates, and proteases. These all the three types of enzymes that are found in our human body as well. They are used to break down proteins, fats, and carbohydrates. For lipids, there is lipase. For carbohydrates, there is carbohydrases, and for proteins, there are proteases. Okay. Now the concept is that if these enzymes can break down proteins, carbohydrates, and lipids in our body, they can also break down these substances when they're outside the body. For example, when you are having a stain of oil in your shirt, so you use enzymatic surf. You also use the enzymes in paper industries. They are also used in the food industry, where these enzymes have a very important role in order to convert the proteins, like carcinogen is converted into casein. So similarly, enzymes, carcinogen is basically a protein that is present in the milk, and when a certain enzyme act on it, it is converted into casein, which is an insoluble form of protein. So that is again used in the food industry. Enzymes basically work by a simple mechanism in which there is a substrate that is recognized by an enzyme, and then they form an enzyme substrate complex, and then the product is formed. The basic thing that you need to know is that the active site is complementary to the shape of the substrate. Okay. Then we also need to know that enzymes are reusable, or they are speeding up a rate of reaction that is reversible in nature. There are two things that we are going to discuss how they affect the activity of enzyme. One thing is temperature and the second thing is pH. In your objectives, it has been mentioned that you should know the effect of changes in temperature and pH on enzyme activity in terms of kinetic energy, shape and fit, denaturation, and the frequency of effective collisions. Let's start with the very basic thing. This is a particular graph which is drawn for the effect of temperature. On the x-axis, I have taken temperature, and on the y-axis, I have taken the rate of reaction. If we talk about the enzymes of human body, in human body, the temperature is the optimum temperature is 37 degrees centigrade. So it's obvious that all the enzymes will work at their level best at the optimum temperature, that is 37 degrees centigrade. After that, the enzymes will start to lower down their activity. In the beginning, they increase the activation energy, they increase the rate of reaction with the increase of temperature because the kinetic energy of the molecule is increasing. But then, as they cross the optimum temperature, the active sites they start to deform in shape. We'll discuss it with the example of an egg. You know that egg is protein in nature, but when you fry the egg at a certain temperature, it loses its shape. It changes its shape. Similarly, enzymes are very sensitive to temperature. They change their active site. The active site gets denatured and deshaped, and they don't bound to it again. Now, if we talk about the pH, now, the pH is again a very important factor that controls the activity of the rate of action of enzymes. In our human body, the pH of the whole human body or different organs is not same. For example, our pH of stomach is 2, which is highly acidic because of the HCl present in it, whereas the pH of our mouth is neutral, and the pH of our small intestine is alkaline. So when the digestion takes place, the enzymes have to work at different pH at different sites. We talk about the pH of the stomach, which is it's highly acidic. So it means that the stomach enzyme won't work in the small intestine. So that's why you can see that for the graph of pH, there is a decline in both sides. I hope you have got the concept. If you have any questions, please ask in the comment section. Thank you for watching. Bye-bye.